This is a production of Cornell University.
Okay, so my name is Alyssa de Villiers. I'm a third-year MS PhD student in the lab of Dr. Carmen Anid Martinez, and I'm really excited to share my research with you today. And I'm going to try using this clicky thing. And here we go. So first, I'd like to acknowledge my committee members, Dr. Nicole Benedek, Dr. Natalie Capiro, and our collaborators, Dr. Christy Goodale and her amazing lab group in ecology and evolutionary biology, and also Dr. Taryn Bowerly in horticulture. And then, of course, the lab group I'm in, all the great members of it, and particularly Anid, thank you so much. I've been lucky to receive funding through the USDA for the first 2 years of my program. That has been really wonderful. And I'm recently funded through the Cornell Atkinson Center for Sustainability Graduate Research Grant. So to give you a general idea of how I anticipate my degree going, the first 2 chapters will be based on my master's project. This involves acidification and nitrogen addition in forest soils and looking at the effects on the water extractable fraction. And then the next 2 chapters that I'm proposing for my dissertation will shift to look at cellulose decomposition and trying to link enzymatic breakdown with this technique that Anid mentioned of density functional theory. So diving right in to the master's project, as I said, this is looking at acidification and nitrogen addition in forest soils. And while our collaborators in the Goodale Lab group are looking at ecosystem fluxes of carbon and nitrogen, and the Bowerly group is looking at below ground carbon allocation and root exudates, we're focused on the soil solution, or what comes off of the soil into water in the soil. And our hypothesis was that dissolved organic matter composition and then absorption to soil minerals to form mineral-associated organic matter would be altered by nitrogen addition and acidification. This is an important issue because, as probably many of you know, we've had acidification and nitrogen addition going on for quite a long time in our soils. So what we see on the right here is global sulfur emissions. And through the combustion of fossil fuels, and particularly coal, we started having an increase in the 1900s. And then what you see, I think I can, here we go. So what you see here is around 1950, we have a sharp acceleration. And down here, this is nitrogen, and in black is atmospheric nitrogen. So this is nitrogen sources in the Chesapeake Bay. And you see that same pattern. So in the 1950s, we have a big acceleration of atmospheric nitrogen, and then we see it peaking around 1980. And so what happened then was essentially the Clean Air Act started regulating sulfur and nitrogen emissions. So on this picture, what we see is that through the combustion of fossil fuels, sulfur dioxide and nitrogen oxides are released to the atmosphere. They can travel, and then through dry or wet deposition, they get distributed across the landscape. And that can cause acidification. It can cause nitrogen addition, which itself can cause acidification. And then also we might have other sources of nitrogen addition like ammonia volatilization or runoff. So as you can see in that graph, we've actually already peaked on those things. Those regulations worked and they helped to bring it down. And this only is showing until 2000 in that top graph, but those numbers continue to fall. So actually what we're seeing now that's more important than the effects of acidification is really what's going to happen as we recover from acidification and recover from nitrogen addition. So we want to see what effects we have on the soil from that. And one of them that can be happening is that more dissolved organic matter can leave the soil, and that can cause lake browning, where the water is getting more organic matter and more color in it. And then that in turn affects the aquatic life in streams and lakes. Also, we are thinking about soil health in terms of losing nutrients or retaining nutrients and carbon in the soil. And climate change, if we have more carbon coming off into the soils, it's possible that microbes would be able to respire that out as CO2, and we might lose that from the system. So that sort of frames what we're thinking about in this project. The setup we used to look at this was actually pretty exciting because it's been happening for over a decade. So before I was involved with the project, these were already treatment conditions that were set up in sites around Ithaca. There's a control treatment with no amendment, an acidified treatment where elemental sulfur was added, a nitrogen addition treatment where nitrate has been added, and an acidify plus nitrogen addition treatment where ammonium sulfate has been added. So the idea there is that some effects might be from both acidification and nitrogen, but some might be from just acidification or just nitrogen addition. So we're going to call each of those treatments a plot. The plot was 40 meters by 40 meters with a 10-meter buffer. And the important thing to know here is that those treatments were each replicated 6 times. So we have 24 total plots distributed at 3 sites around Ithaca. And in general, these are forested hillsides that are silt, loam, and septisols. But there is, there are some pretty big variations in organic matter and pH at these different sites. So we do take that into account when we're doing the statistical analyses. At each of these plots, soils were sampled in 5 locations and composited to the plot level and then sieved, and 4 depths were taken. So we have the forest floor, which I'm going to call the O horizon, and then pretty shallow depths, so 0 to 3 centimeters, just the first top inch of soil, 3 to 10 centimeters, and 10 to 20 centimeters, which all fall into the A horizon mineral soil. And down here, in this graph, this is just to show that we did have acidification in the treatments we expected to acidify. So acidify only brought the pH of the forest floor down to an average of 3.5. So this is really quite acidic. At this point in the study, these soils that I'm looking at are from 2022. We only saw a small effect of pH in the acidify plus N condition. So that's also important to note. And then once we look at the 0 to 3 centimeters top inch of soil, both acidify plus nitrogen addition and acidify are bringing the pH to around 4.6. So I just want to ground everybody in what kind of pHs we're talking about, because some of them are really quite acidic. Okay. So I was not involved in the soil sample collection. And then these samples, as I said, were composited, sieved. They make it to me. So here's where I come into the story. I receive that soil and I shake it with water and then filter it to less than 2 microns. So now what we have is this water-soluble fraction. So everything that's going to come off the soil into the water, sometimes we might think of this as the dissolved organic matter. And our first objective is to find out what's in that. And as many of you will know, it can be very hard to find out like the exact compounds that are in there. There's thousands of compounds. But we're going to pick some analyses to give us an idea of what's in there, and that's that characterization step. Then, if you think about that water moving through the soil, and now it's picked up various different things into it, now it's going to start interacting with the soil around it. So the next part to simulate that is a batch experiment, where we take that dissolved organic matter and shake it with minerals that are found in the soil and see what sticks. So the assumption here is that you have some amount of, say, carbon or nitrogen in solution. You shake it with a mineral, you remeasure the solution, and whatever amount that's decreased, that has been sorbed or stuck to those soil minerals. So that's part 2. And then I've completed the experiment, but not this variable importance, which is going to be the next aspect. And the idea there is to say, what factors are controlling how much carbon specifically is sticking to these different soil minerals? And then finally, objective 3 is ATR, FTIR flow-through experiment. And the important thing to know here is we're going to take a soil mineral, we're going to add some organic matter on top of it. That's going to create what we call MAOM, or mineral-associated organic matter. And then we're going to run root exudates off of that and see what happens. So root exudates can push off organic matter, possibly off minerals, or maybe they'll get stuck themselves. So those are the objectives. And one really important thing to remember the whole time I'm talking, I think probably people in this room mostly already know this, but I'm not talking about bulk soil or the carbon and nitrogen in the soil. I'm talking about the carbon and nitrogen in the water that comes into contact with the soil. And so I might call this the water-soluble fraction. I could call it the soil solution or the aqueous phase, or I might talk about soluble carbon and water-extractable nutrients, but just remember that it's all in the water. And why we care about this fraction is because it's highly mobile, it's responsive to environmental changes, and it's available to plants and microbes without a lot of additional effort on their part. And why I have all these arrows is not to say, oh, I've gotten every single input or output of the system. It's just to illustrate that if we find that we have more or less of something in that water-soluble fraction, it could be for a number of reasons. Maybe it's because we have more inputs. But equally, it could be because we have fewer outputs. So it kind of, as usual with soil, is a mess for trying to determine exactly why something's happening. But just keep that in mind for when we see an effect. So jumping right into the results, I'm going to start in the O horizon and talk about dissolved organic carbon. So here on the left, we see that all of our treatments reduced the carbon coming off the soil at this point, which is 10 years, a decade into the study. So then we want to see what can we say about that carbon. So just saying the amount of carbon is pretty general. And one measurement type we can get is aromaticity. And aromaticity you can think of as, sometimes people think of these benzene rings. So it's where you have bonds where electrons are shared, and it tends to make the molecules more stable. And also, if it is a complex system, then it can add more color to those molecules. So what we see here is that the only treatment that had an effect was our acidification treatment. And that lowered aromaticity of the dissolved organic carbon. Now moving to this graph, this is sort of a proxy measurement for size or molecular weight of molecules in solution. And what we find is acidification alone is the only condition that had an effect. And its effect is, if we follow this arrow up, to make molecules smaller. So we start to get this picture, as we can see down here, if we imagine these little brown balls as dissolved organic carbon, that when we acidify, what's coming off the soil is less in amount of carbon, it's smaller, it's less aromatic, and I'm not showing the box plot here, but it also has a trend of less color. At the same time, when we add nitrogen, we still see less dissolved organic carbon, but no change in size or aromaticity. And at first I thought, oh, the nitrogen's having a modifying effect on the acidification. But remember that in this O horizon, that acidify plus N treatment actually wasn't that acidified. So I think based on the fact that size, aromaticity, and color in the O horizon have a linear correlation to pH, what we're actually seeing is just an acidification effect. So why might this be happening? And this is where we get into, I'm just giving you some ideas on this, and everybody else is welcome to add their ideas in at the end. So there could be biotic and abiotic reasons. Why might we have less input of DOC into our water-soluble fraction? Well, with nitrogen addition, it's been shown that there's fewer root exudates coming into the system. Essentially, the trees are getting enough on their own of their requirements met that maybe they don't need to be sort of enhancing the system to their benefit in that way. Alternatively, maybe we're retaining more of the DOC on the soil. So when you have acidification, that is going to take oxygens with negative charges that are in solution and add a proton to them. So now they're not going to be negatively charged. And that tends to reduce repulsive interactions between organic interactions we'd be talking about in the O horizon. And then that could mean that more is retained and less is coming off into solution. Okay, so then in terms of the smaller size in the acidification treatment, a biotic reason might be that at that really low pH of 3.5, we could be inhibiting microbial decomposition. So maybe a little bit is happening, but they're just knocking off a few number that are just small bits off the end. And then an opposite idea would be that, in fact, larger molecules are coming into solution, but because of that acidic pH, they're broken down into smaller molecules rapidly, abiotically. So this is the type of thought process we're going through when we're doing this. But overall, the implications, and this is really something that other studies have found also and is really important, is that as we recover from acidification, the opposite effect can happen. So that means that we would have more DOC coming off soils, but also that it's made up of larger and more aromatic molecules with more color. And so why that matters is that idea of lake browning that I mentioned earlier. So if we have more DOC coming off, and also it's more aromatic per unit of carbon, that can impart more color to waterways. And they do actually, that is something that other people are finding and researching actively. But because we see that nitrogen addition also reduced the total amount of dissolved organic carbon, it seems like that the recovery from nitrogen addition could add to the effects of the recovery from acidification. Okay, straight from carbon to nitrogen, all the important players here. So in this top left box plot, we see total dissolved nitrogen, and acidification plus nitrogen is reducing total nitrogen, and acidification has a trend of reduction. All right, so then we want to think, well, what kind of nitrogen are we talking about? So first we're looking at inorganic nitrogen, nitrate, and ammonium. And we see something pretty interesting, although not wholly unexpected. Here in purple is the acidification only treatment. And what you see is that this is for nitrate in that graph. So what you see is in the O horizon, there is no water extractable nitrate at all coming off of these soils under acidification. And when you go down into the top inch of soil, there's still almost none. And then we don't see an effect for the other treatments. And then on this graph, this is ammonium. And what we see is that there's a little increase in ammonium under acidification and a decrease in ammonium in acidification plus nitrogen addition. And that's kind of interesting. If we're adding nitrogen in that condition as ammonium, you might think, okay, we're adding ammonium to the soil, ammonia might go up. But that's not what we see. So here, pictorially, you can see that the ammonium in acidify, it's mostly ammonium, 0% nitrate. And here in the acidification plus nitrogen, we have a little reduction in ammonium and increase in nitrate. So why might that be happening? I think one of the major bacteria that we need to think about when we talk about ammonium and nitrate is nitrifiers. And probably there's at least some role going on here for why we're seeing that. So there could be a number of reasons that we're losing that nitrate in acidify, but a microbial reason would be that even though nitrifiers can operate at some acidic pHs, again, we're really acidic. And it's possible that we're seeing inhibition of nitrification. And so the ammonium is accumulating and not being converted to nitrate. But we also could have more output. And in fact, I'm sure these two are working together. So from abiotically speaking, nitrate is an anion. It's not going to stick to negatively charged soil particles. So you're just going to have loss from the system. And as acidification pushes out base cations and those leach through the soil, nitrate is a good kind of companion to exit with them. Also, we could have more output due to chemodenitrification or self-decomposition of nitrate. And then I'm actually going to talk about this one at the same time because this is still in the acidification treatment. So as we add protons or hydrogen ions to the solution, that might also push off sorbed ammonium. So those are some reasons that would explain what we see in acidification. How about in the acidify plus N? Why would we have that decrease in ammonium? Currently, my idea is that maybe we have an increase in nitrifiers, and they're able to process that ammonium. We're saying, here's your substrate, and we're adding it every single year. Here you go. And maybe they're pretty good at processing that and continually putting out nitrate into the system. And you really need that kind of continuous input of nitrate because, again, it's not going to be retained in the soil. It's either going to be taken up by plants or leach out. And what we've seen is that water extractable nitrate was completely absent after a decade of acidification. And it's not the nitrogen addition, but the acidity that is the major factor influencing nitrogen speciation. Okay, so now we're going to move out of the O horizon and into the mineral soil. And we have a new player in the game, and that's aluminum. So what this first graph is showing is the relationship between aluminum and dissolved organic carbon. And this is a known relationship. So again, not wildly surprising, but I think the important thing to emphasize here is we don't see a relationship in the O horizon. So aluminum's really not playing a role, even though we have those really acidic soils in the O horizon. Then when we get down even an inch down, all of a sudden we see that correlation. So moving to this graph on the upper right, this is showing size or apparent molecular weight on the y-axis to aluminum on the x-axis. And again, what we're seeing is that aluminum's really not affecting the O horizon and very, essentially not in the first inch of soil. But once we get down a little deeper to 3 to 10 centimeters, as we increase in aluminum, molecules are getting larger in size. And so a lot of times people talk about cation bridging, which is the idea that a positively charged particle with more than one positive charge could essentially mediate between two negatively charged carbon-containing molecules. So if you have two carboxylic acids with negative charges, and you have, say, a calcium with a charge of +2 in the middle, it's kind of making everyone play nice, right? It's making it so that you can have a little more, a little less space between those negative charges. And aluminum is a +3 ion, and we could expect that the same thing might be happening. So this could be evidence of that. Then on this bottom graph, this is showing aromaticity again, and there is a relationship between aromaticity and size in the O horizon and the top inch of soil, but it's pretty flat. You can see that here. But then once we get to 3 to 10 centimeters, we see a steeper slope, and that's saying as we have more aromatic molecules, they're larger. So we start to get this picture where there's more aluminum in solution, more carbon in solution, there are larger molecules with more aluminum, and we have these larger when there's more aromaticity. And so I better click along here. So why would we care about that? So the next part is leading into the batch experiments. So once we have an idea of what's maybe making larger molecules, which tend to sit down on mineral surfaces more, and we get the sense that aluminum is playing a role in that, and we get a sense of how much carbon we have in solution. Now we want to know, how is it going to stick to the rest of the soil? And so for this, we move to objective 2, which is the batch experiments. And so I extracted dissolved organic matter from the 0 to 3 centimeter layer, and then shook it with goethite, an iron oxide hydroxide, kaolinite, a 1-to-1 aluminosilicate clay, gibbsite, an aluminum hydroxide, and the 3 to 10 centimeter soil, which I'm not going to show the results of right now. But this graph, which is, again, I'm only going to show one result from this. We're still processing the data, but this graph I think is kind of cool. This is aluminum sorbed on the x-axis and organic carbon sorbed on the y-axis. So if we just look at the amount of organic carbon sorbed to the soil, we see, as expected, kaolinite is really not sorbing very much carbon. And then gibbsite and goethite are sorbing, at least in the same order of magnitude. And the interesting part is we saw that aluminum association with carbon in the solution, but now we're still seeing it on what's been sorbed, so what's left the solution once we shake it with those minerals. And there could be a couple reasons we might be seeing this. So this bottom one is saying here's aluminum associating with organic molecules in solution, just like we saw maybe on that previous slide. And then this is a chemosorption to the iron and goethite, and it's sort of all helping it come together, kind of co-sorbing together. Or alternatively, maybe aluminum first sorbs to the goethite and then allows for more organic compounds to pile on. So either way, we're going to be investigating what's going on there and seeing whether we can see, particularly we can look for this bond here of iron, oxygen, aluminum. So that's something we'll be continuing to do. All right. For now, that's going to wrap up the master's project discussion, and I'll move on to what I'm proposing for my dissertation proposal. While this is still relevant in forest soils, I'm actually mentally shifting more to agricultural soils. I think that we talk about soil health and nutrient cycling and how to build soil health, which is something really important to me as a small farmer. And one thing we notice is one way is to add plant matter and nutrients. So what does all that plant matter have? It has cellulose. And so how that cellulose breaks down and how it interacts, how quickly it breaks down, is going to affect that nutrient cycling through the system. So I'd like to look at 5 factors on cellulose decomposition: pH, enzyme and cellulose concentration, the presence of 3 soil minerals, probably the ones I just showed, the presence of low molecular weight organic molecules like amino acids, and then the presence of a divalent cation. And in agricultural systems, calcium is probably the one we'd want to look at because obviously when you're liming, you're adding calcium to soils. So the question is, could these different factors affect how quickly cellulose is cycling? And the sort of inspiration for this comes from the way cellulose enzymes work. So here, this is cellulose. It's made up of chains of glucose monomers called fibrils that are then hydrogen bonded together in this actually rigid crystalline structure. Then to break it down, you need 3 different types of enzymes: endoglucanases, exoglucanases, and beta-glucosidases. And this is an example of the exoglucanase cell 7A, and I just think this is so cool. This is a molecular dynamics simulation of the, oh, hold on. Whoa. So that's a simulation, a molecular simulation of that enzyme at work. And what we see is that here, it's picking up a fibril off the cellulose surface, and it's going to be knocking off 2 and 3 chunks of cellulose at a time off the back end here out of the catalytic domain. And then it has this flexible linker, and in the front it has a carbohydrate binding module, which they sometimes call a foot, and that's anchoring that enzyme to the surface. So that's a really important part of the whole thing. And in this foot, we can see it zoomed in over here. It's got 3 amino acid residues that are helping it bind, and those are tyrosine. So the question is, this is going to be processing along the cellulose surface, breaking down these little units as it goes. What happens if it hits an obstacle? And so when we think of absorption and protective absorption, some ways that we might see that for the cellulose enzyme would be the enzyme could bind to a mineral and then not be able to get to cellulose and break it down in the soil. Or maybe the cellulose could bind to a mineral and then not be available for the enzyme. But another type that I wanted to investigate is, what if something binds to the cellulose? Like, what if a couple tyrosines come in here? Could they bind? And would that then block the process of this enzyme moving down the surface? So to test that, I have these 8 different treatment conditions, and I'm going to shake each of them with cellulose to allow any interactions that are going to happen. And then we'll add an enzyme mix from a fungus called Trichoderma raceyi that has the ability to break down the cellulose all the way to glucose. So then we can measure glucose as a function of time and see how much cellulose is being broken down. And the idea would be, we assume that in some of these conditions, you'll find protective absorption of different types, and then we can investigate what we see there. Okay, and then finally for objective 2, I'd like to link this technique called density functional theory and see if we can get it to be predictive in a slightly more macro scale of enzymatic breakdown. So what that might look like is, in density functional theory, which is based on quantum mechanics and models electron configurations, we can model a surface, like a mineral surface or the cellulose surface, and then we can model a molecule like an amino acid, and then we can put them together and see how they bind. And so the question is, and I don't know the answer, but can we, if we can accurately, by corroborating it with laboratory experiments, if we can accurately predict which molecules are going to bind more strongly, can we use that to predict whether or not, say, an enzyme is going to have trouble breaking something down? And so that's the idea with this. And why it's exciting is partly because it's high throughput. So once you model your surface, you can model a lot of different molecules, although again, you have to corroborate the results with your laboratory results. And then there's this question, could it be predictive? That'd be pretty exciting if we could measure a cellulose surface, measure how tightly something bound to it, and that correlated to how efficient the enzyme was. I'm not sure that would be possible, but that'd be exciting, I think. So that's everything I wanted to share with you today, and I really appreciate everybody coming, and I'd like to open it up for questions. Johannes.
Yeah, thank you very much, Alyssa. Great data and wonderfully presented. Congratulations.
Thank you.
Just some nitty-gritty questions.
Yes.
I wanted to learn from you. You said you're filtering your DOC at 0.22. Why that? Soil scientists usually do 0.45, aquatic scientists 0.7. Why 0.2?
Well, I mean, I think that the goal with all the filtering is to make sure you're not getting like microbe sterile, so not getting microbial biomass, but also not getting like fine soil particles.
Mm-hmm.
So I think that we just chose 0.2 to make sure we weren't getting anything in that range. I think there are some things between 0.2 and 0.4, particularly like colloidal
Yeah, no,
bits.
for LCMS, we're doing 0.2.
Two, yeah.
So I was just wondering whether there was some other reason that I wasn't.
No, it's just that reason, and I'm trying to remember the chart of the different sizes, but I remember at some point looking at it, and that's what we chose.
Yeah, no,
Yeah.
It's always interesting how the different fields are using different cutoffs.
Right, so you might lose a little
Technologies.
bit that's in that range of 0.2 to 0.4, yeah.
Yeah.
Did you have another one?
Yeah,
Yeah.
a few ones. So did you look at, do you think sulfur plays a role in all of that? I mean, you honed in on the nitrogen and
Yeah,
acidity,
yeah.
but then there's sulfur as well. And I know that you fundamentally change when you add sulfur.
Yes.
And also with the historic emissions, that it changes organic sulfur forms.
Yeah, I think
Does that play a role in something?
I'm sure it plays a role.
Yeah.
Yeah, and sulfur can be involved, there's redox reactions going on there. And in fact, when we add the elemental sulfur, it's primarily acidifying by microbial oxidation. So yeah, it's a lot of sulfur cycling going on. And unfortunately, we were not well set up to measure the sulfur in solution. So I don't have a lot to say about it, but that's definitely an important role. And I do know some other papers are looking at like sulfate concentrations also.
That'd be the counter ion for your.
Yeah, it could. In fact, what could be a counter ion? Primarily nitrate and sulfate. So really good
Yeah.
point. Yeah. Yeah.
Thank you.
Yeah, of course. Yeah, Jonathan.
I got a question about the aluminum component.
Yes.
Particularly since you're acidifying,
Yes.
you're going from this gibbsite into these ionic state aluminums, but with the pH, you're increasing. As you decrease the pH, you're increasing
Accelerating aluminum. Yeah.
concentrations. I'm wondering, certainly from a concept of recovery from
Mhm.
acidification, what's the tag there that this is what we're going to see is recovery, rather than this is just more. We're not. I mean, it's no more acid rain in a sense,
Right.
because of the EPA, but still.
I actually think that's something I'm really interested in thinking about. And I don't know if other people have this problem, but I find my mind can go round and round in these sort of different circles of, oh, maybe it'd be this way, or maybe it would happen this way. But it depends what role aluminum's playing in the absorption. So we're not seeing it in the O horizon. And a lot of the research out there that's similar, that's looking at that runoff into streams and lake browning, is talking about what's coming from the O horizon. So
Yeah, but the O horizon is not going to be your source of aluminum.
exactly, right, exactly. So that's interesting. So it depends. Is that going to be going into the soil and then interacting with aluminum, or is it just running off the surface? But if it's in the soil, I would expect that what we're seeing is more aluminum is actually helping it stick around. So I would expect that as the pH goes up, that it would reduce aluminum, and that could also exacerbate losing, more carbon. That's the way I was looking at it
So
anyway.
you're thinking it's more like a spodosoil than the inceptisoils that we have around here. I mean, there's that experiment that's up on Turkey Hill or Game Farm, well, not Game Farm, up to Mount Pleasant, that they actually put the trees in place
Okay.
to see if because it's the O horizon
Yep.
using the acids.
Yep.
If the organic material that's coming from these trees are actually creating the spodec horizons, which would then
Okay.
be these BHs and BHSs farther down. And when I looked at those numbers that you
Yeah.
were saying, this is the A horizon, those aren't A horizons. You got an A horizon that's maybe this thick, and
Yeah, they
get down
consider like the soil series, they consider it AP. I don't know. I don't know. Depends how you want to think about it, maybe. Yeah. I mean, I think it's all effectively been disturbed soil at some point. So, but I do think that thinking about how the recovery acidification will affect the aluminum is important, and whether something like calcium or a different divalent, cation could sort of take over some of that bridging role would be interesting, but not something we're at yet, I guess. Yeah.
Thank you.
Yeah, of course. Any other questions?
No? Okay, so then round of applause, and I'll see you next time.
This has been a production of Cornell University, on the web at cornell.edu.